When the fungal pathogen Phytophthora infestans attacks a tomato leaf, its zoospores actively breach the plant's thick cell walls. Once inside, they multiply rapidly, causing the devastating cellular decay known as late blight. Agronomists face a severe biological limitation in fighting this disease. Specific resistance to late blight race 1 is completely absent in current commercial tomato cultivars. Traditional crossbreeding strategies require years of generational selection to introduce new traits. This timeline is entirely too slow to keep pace with the rapid, seasonal mutation rates of aggressive fungal pathogens. Instead of waiting for field generations to slowly adapt, researchers can use a controlled laboratory environment to intentionally force rapid genetic adaptations at the cellular level. To prevent total crop failure, the agricultural sector must pivot to these accelerated, non-traditional methodologies to generate pathogen resistance on demand. Tissue culture is typically utilized as a stable method for cloning plants. However, removing cells from their natural developmental cycle and placing them into an artificial in vitro environment subjects them to severe chemical stress, creating a highly volatile and mutagenic state. This stress triggers somaclonal variation. This mechanism generates rapid, exploitable genetic diversity entirely from the plant's existing genome, avoiding the use of transgenics. This schematic flowchart outlines the chronological steps of the in vitro methodology. Step 1 is explant isolation, the precise removal of target tissue from a sterile donor plant. Step 2 places that tissue into chemical media, forcing the cells to de-differentiate into an unorganized mass known as callus. Step 3 manipulates the hormone levels in that callus, compelling the unorganized cells to regenerate into a viable physical plantlet. Step 4 involves maturing that regenerant into an adult plant and self-pollinating it to produce the mutated generation of seeds, called the R1 progeny. Step 5 takes that R1 generation and aggressively screens it against live pathogens to isolate any mutants developing resistance. Tracking a 1999 methodology by researchers Chowdhury and Shah demonstrates exactly how controlling the initial explant environment dictates the success of this entire mutation pipeline. The researchers began with strictly controlled starting material, sterile tomato seedlings grown exactly to the 13-day mark. To test how the physical origin of the tissue affects the culture, they excised precise 5mm segments specifically from the seedling's cotyledon, the embryonic first leaves. They then isolated the tissue variable by making identical 5mm cuts from the hypocotyl, which is the pale stem located directly below those leaves. Both tissue types were placed onto nutrient plates, supplemented with specific growth regulators, zeatin and IAA. This split bar chart compares survival rates of the two tissue sources after 4 weeks. Cotyledon explants heavily dominate the survival metrics. At 8 weeks, this advantage compounded. Cotyledon segments drastically outperformed hypocotyl segments in regenerating physical shoots. This morphogenic disparity is explained by the hormone hypothesis. The varying success rates are caused by the uneven distribution of endogenous growth regulators already inherent to the specific anatomical regions of the donor plant. Cellular regeneration competence is not uniformly distributed throughout the plant. It is strictly dictated by the developmental stage and exact physical origin of the cultivated tissue. Beyond the physical origin of the tissue, the plant's underlying genetic identity establishes the ceiling for success. The researchers evaluated specific tomato genotypes with known disease profiles, establishing them as either resistant or susceptible to baseline pathogens before culturing. This chart displays average shoot regeneration rates at 8 weeks across different genetic lines. Genotype LA1623 exhibited high-frequency regeneration, emerging in just 2 weeks, beating the standard 4-week period. Conversely, genotype Nova suffered severe failure, yielding less than 1 shoot per explant. Nova represents what is known as a recalcitrant genotype, a genetic line where standard in vitro hormone treatments completely failed to trigger morphogenesis. Overcoming this requires highly specific protocol adjustments. For example, genotype LA1456 initially showed low regeneration, which was only corrected by harvesting tissue from younger donor seedlings. In vitro breeding protocols cannot be standardized into a universal formula. Successful mutation induction requires strict, bespoke calibration to the precise genetic profile of the target cultivar. Once the raw shoots are generated, they enter a critical transition phase. They are excised from the hormone-rich media and transferred to a restrictive environment designed specifically to induce rooting. Over the next 15 days, isolated shoots must undergo a severe morphological transition, building an entirely new, viable root system capable of supporting independent life. These highly vulnerable regenerants are then subjected to a delicate greenhouse acclimatization process, gradually transitioning them into mature, seed-bearing adult plants. Surviving the extreme physical and chemical stress of this specific morphogenesis pipeline is the exact trigger that primes the plant's genome for exploitable epigenetic shifts. The researchers collected seeds from the self-pollinated regenerant plants, generating 48 distinct R1 somaclonal families for analysis. Physical evidence that genetic alteration successfully occurred materialized immediately. As the R1 seeds grew, clear physical anomalies, termed early mutants, appeared in the seedlings. Some lines presented lethal mutations. Clone 115, for example, emerged completely albino and rapidly failed due to structural deficiency. Other lines displayed non-lethal morphological anomalies, growing distinct, abnormal pot leaf structures during their early growth phases, instead of wild-type foliage. With the mutation rate visually confirmed, the researchers moved to the final stress test, challenging these mutated R1 families directly with late blight zoospores to uncover any hidden biochemical resistance. They prepared a highly concentrated zoospore suspension utilizing the virulent isolate Ca65, meticulously calibrated to 10,000 spores per milliliter. The mutated seedlings were sealed inside strictly controlled, clear plastic propagator environments and aggressively spray inoculated until the suspension ran off the leaves. Because the tissue culture successfully induced obvious physical changes like albinism, it proved the environment was volatile enough to potentially generate the invisible cellular disease resistance the agricultural industry requires. This line graph tracks seedling survival percentage over 14 days of pathogen exposure. By day 7, the unmutated control group of LA1623 takes a steep plunge, dropping to a mere 20% survival rate. By the 14-day mark, the control group experienced complete cellular collapse, resulting in a 0% survival rate. When compared to the control group, the data for clone 123 diverges, stabilizing and remaining elevated throughout the 14-day trial. This shaded delta reveals the survival gap. Where the control group reached 0, clone 123 maintained a 45% survival rate against the Cas65 isolate after 14 days of exposure. The parent genome of these plants possessed absolutely no defense against tomato race 0 of this pathogen. By forcing the tissue through the in vitro pipeline, the researchers engineered stable, spontaneous genetic resistance entirely from within the plant's own biological architecture. Systematic induction and screening of somaclonal variation offers a non-transgenic pathway to generate stable resistance from existing germplasm, providing a methodology to outpace the mutation rates of aggressive pathogens.